we're going to be talking about is growing bacteria in a lab, how we count bacteria in a lab, how we maintain cultures in a lab, and make sure that the bacteria are happy and healthy growing in a lab. It seems like it should be a fairly straightforward thing to do. You just give them food, put them in an incubator, things like that, but it's not as always as easy as it seems. So one of the first things you need to understand about growing bacteria in a lab is the concept of nutrition, and that's feeding bacteria or feeding living organisms for that matter. Living organisms, for the most part, if they're not autotrophic, meaning they make their own food, they're going to acquire nutrients from outside of their body, outside of their cells. So nutrients are things that are going to be absorbed from the environment in the form of like proteins and lipids and sugars and things that your cells will bring inside of the cell. They'll break them down and change them around and build them up into different types of sugars, different types of proteins, different types of things that the cells will then use for their own survival, making energy for the cell or making repairing the cell, making new proteins that the cell needs and things like that. You guys are probably familiar with this concept of something called an essential nutrient. It's something that you absolutely have to have in your diet. If you don't have it, you will be nutrient deficient. You might have a couple of problems making proteins and things like that. Your body might not be as happy as it could be or as functioning as good as it could be. So essential nutrients are things like iron, proteins, manganese, zinc, copper. You got to have a little bit of that kind of stuff in your diet. Things that you absolutely have to have. I mean, you can break those essential nutrients up into two smaller categories again, one called a macronutrient and one called a micronutrient. So a macronutrient is something that you need in large amounts in your diet, things that play really important day-to-day roles in the life of your cells. So proteins, fats, sugars, things like that, carbohydrates, things that your cell needs to make lots of, to have a bunch of them on hand, to be able to make more of when old ones are broken, old ones are used and degraded, and things like that, that your body needs to replace quite often. On the other hand of that, there's something called a micronutrient, things that your body just needs a little bit of. Not too much of, just a little bit. Maybe you don't need to make a ton of this particular type of protein, so you just need a little bit of a particular nutrient for that micronutrient. Or a lot of these things function as coenzymes, cofactors on enzymes, vitamins, things like that you just need tiny little amounts of for your cells to be happy and healthy. So the other concept on this one is organic versus inorganic nutrients. So if you guys recall from chapter 1 or lecture 1, I should say, you guys will recall that organic nutrients, organic compounds have carbon and hydrogen bonded together. It's their definition. A lot of these things are usually produced by living organisms, whereas inorganic nutrients tend to have a lot of other atoms other than carbon and hydrogen bonded together, and they tend to not be produced by living organisms as often. So you can break bacteria and pretty much every other living group of organisms up into how they get their food from the environment. So are they going to be making their food, or are they going to be eating something else to get the food that they need? And so you guys are familiar with this term, with this concept. If you think about photosynthesis and things that are photosynthetic, making their own food. So something that's photosynthetic that can make its own food is called an autotroph. Auto meaning self, troph meaning feeding. Something that can't make its own food is called a heterotroph. Hetero meaning different, different feeding, so a different food source. Most bacteria on the planet are heterotrophs, but there are a couple that can make their own food. Things like cyanobacteria, blue-green algae is another name for that. They are photosynthetic. It's a very simplistic form of photosynthesis, but they are capable of making their own food. Bacteria that make their own food, that or acquire their own nutrient source, you can break that category up again into how they get their sources of carbon, where they get their sources of carbon from. Regardless of if you make your own food through photosynthesis or you eat another source of food to get the nutrients that you need, you still have to get carbon from somewhere to be able to make the sugars that you need to function. Where that source of carbon comes from gives you one of these categories. If you are a photoautotroph or a photoheterotroph, You're going to be getting your light energy from the sun, and you get your CO2, your carbon source, from other things that you eat. Out from usually these things will bring in carbon dioxide from the oxygen and things like that, things like plants. Chemoautotrophs, chemoheterotrophs, chemo meaning chemical. We happen to be chemical chemoheterotrophs. So these things will get their CO2 from some sort of non their energy, excuse me, from some sort of non-light based structure. So they'll use chemical means to make food. Photoautotrophs use light to do that. Chemoautotrophs use chemical reactions to build up food. So that's kind of the difference between them. So we kind of covered this concept on the last lecture when we talked about the concept of fastidious organisms, bacteria that need essential nutrients added to their media, added to their agar when you try to grow them. So if you try to grow something like a streptococcus organism in a laboratory, these guys like a lot of iron in their diet. Nutrient agar doesn't really have a lot of iron in it. So if you choose a nutrient agar plate to try to grow streptococcus species on, they don't have enough nutrients in there. There's not enough iron. These bacteria will grow very slowly, very small colonies. It takes them forever to get any size that you can really function with them if they grow at all. So if you want bacteria like streptococcus to grow in a lab, you have to give them the essential nutrients that they need. You have to give them iron or a source of iron in their media, in their growth platform, the house that you choose for them, their petri dish. So organisms that require these type of added nutrients are termed fastidious. So let's go over that. So heterotrophs. Heterotrophs come in two different forms. You guys are familiar with this concept, especially when it comes to um, us. We are heterotrophs. We eat food. We are chemoheterotrophs. We break down our food through the concept of aerobic respiration. We breathe. We bring in oxygen. We break down food through metabolism. Oxygen uses a terminal electron acceptor to get rid of all the excess carbon dioxide and all that stuff, and blah blah blah. You guys kind of brief run through of metabolism. Well, when you're talking about bacteria, they're either going to break down food that way, or they're going to use one of these two sort of ideas to break down food. So they're either going to break down food the way that we do, just traditional means, or they're going to be one of these guys, something called a saprobic bacteria, a saprophyte, or a parasitic bacteria. So saprophytic bacteria, or microorganisms, tend to be fungi and things like that, tend to be saprophytic. What they do is they release toxins from their cells or from their root system. For fungus are called hyphae. They will release digestive enzymes from their cells. Those digestive enzymes then go out into the environment, break down whatever the organism is sitting on, and then they resuck up that pre-digested nutrients. So parasites, on the other hand, parasites tend to absorb nutrients from their host. So the host is the nutrient source. The host tends to already have broken down the nutrients, things they steal from your intestines, like tapeworms and stuff. They steal the food that you've already broken down, or they steal blood from you and things like that. So they tend to steal the food from you. So you are their source of food in that case. So moving on to growing bacteria in a lab. Bacteria in nature can be found growing pretty much everywhere you look. They can be found growing on the deepest ocean floors down here. This is at the bottom of the Marianas Trench. This is about 2 or 3 thousand. This is about 2 or 3 miles below the ocean floor. I think it's actually deeper than that. What you'll see here are these little vents. They're underneath the ocean, and this is not smoke. This is pure hydrosulfuric acid. It's black hydrosulfuric acid coming from the center of the earth. It's so deep down in the core. The temperature of the earth down there, the temperature of the water is over boiling. The pH of this is about 1, so it's highly, highly acidic. Bacteria love it. There's no light down here at all. These guys use total chemical means to make all of their energy. We would die simply from the pressure of the water alone. Bacteria love it. If you come over here, this is a very, very, very freezing glacier. You guys can see this black smear here, this kind of stuff. It's not dirt. It's bacteria that grow on the surface of a freezing glacier. So different species of bacteria can be found everywhere in every environment and everywhere in between on different surfaces of the Earth. And it's probable that they're found on other planets too. They're just quite small, and we haven't really been able to look for them yet. So, given the fact that they grow in all these different environments with all these crazy conditions going on, it's kind of difficult to replicate something like this in a lab or something like this in a lab. If you want to grow these bacteria happily, you have to give them boiling hot water with an acidic pH. Or over here, you have to be freezing with tons of ice. Both of those are quite difficult and a little dangerous to replicate in a lab. So that creates a bit of a problem when you try to grow bacteria species in a lab. We cannot grow most bacteria species in a lab. We're kind of limited on the ones that we can grow. So one of the first things that you need to understand about growing in bacteria in a lab is how bacteria replicate in the first place. This is a very easy concept called binary fission. This is asexual reproduction in bacteria. They are capable of a form of sexual reproduction, which we'll cover later. So essentially how this works, you start out with one bacterial cell. Their little bacterial chromosome inside, also called a nucleoid. The little bacterial cell starts to grow and become large enough that it is ready to divide. It doesn't need sexual reproduction. No mate is required for this. Essentially what's going to happen is the little bacterial chromosome will replicate itself. The cell will make another copy. So you got two copies of the original chromosome in there. Those chromosomes will then move to one side of the cell. You guys can see that here. And then the cell will just split itself in half through the process of cytokinesis. So then you've got two identical copies of the original cell here. Very, very, very simple concept. Very easy. Very quick way to replicate. But the problem here is it doesn't create any genetic variability. This guy is identical to this guy. So if a disease will kill the original cell, it'll kill both of these. But whereas something that undergoes sexual reproduction, the genes are shaken up a little bit. There's a bit of a chance that this guy might be immune, whereas the parents are not immune to a particular disease. Where in these guys, they're all identical, they're all the same. It's going to cause some problems, although that does allow for easy testing in the sense of everybody's the same in a colony. So when you're growing bacteria in a lab, every bacteria is essentially going to follow the following pattern of growth. They make it's pretty easy to define how bacteria grow in a lab. But one of the first things that you want to understand when bacteria start to divide is something called the generation time. We'll get to that in a second, but let's go over how they grow in a lab to start with. So this is the word form of what I'm about to go over right here. All this in this PowerPoint. So I'm going to work over the graph phase. If you guys needed a word explanation, it's right here. So essentially how this works. Every single bacteria on the planet will undergo this essential type of growth when you introduce it to a brand new colony, a brand new petri dish, a brand new host, aka you get sick for the very first time. So the very first thing that happens right here, this is the number of cells that are found in a sample, and this is the amount of time on this side over here. So the very first thing that's going to happen is bacteria are going to be introduced to a brand new environment right here. You've introduced them to a new petri dish, you've gotten sick, someone's coughed on you, something like that. The very, very, very first time that bacteria is introduced to a new environment, that number will not change. So the bacteria that are introduced there are not going to instantly start to grow and divide. They're going to have to kind of figure out what's going on. Where are we at? Are we in the host that we want to be in? Do we have nutrients that we can use? Can we use the food that we have available for us at all? Is this the temperature that we like? They're going to have to start to figure out what's going on. And given a little bit of time, they will eventually figure out what they've got available for them. They will start to utilize the nutrients available and start to divide. So down here, there's no active division at all in the lag phase. The bacteria are just figuring out what's going on in the environment. So right here, they've started to figure out what's going on, and they're starting to actively divide. They're going up, they're dividing. The number of cells is increasing. They're starting to divide and grow over the size, the petri dish as a whole. They're starting to cause you as a person to get sicker. As they grow and grow and grow, your symptoms are getting worse. This is something called the log phase or the exponential growth phase. The bacteria are growing at a very rapid pace. So eventually what's going to happen is they're going to reach something called the stationary phase. Now I like to explain the stationary phase kind of as the concept of an apartment building. Everybody has to have an apartment. There's only 100 apartments. More than 100 people trying to live in 100 apartments is going to cause problems. Somebody's going to have to share their resources. If 100 people live in 100 apartments, everybody's got the same space that they need, everybody's got the same plumbing, everybody's got the same amount of food, everybody's happy. But if 101 people live in the apartment building, someone's going to have to share, which means someone's going to be deprived of half of the resources that they originally had, which is going to detrimentally impact their lifestyle. They might die. This is something called the carrying capacity. The environment can only support a certain number of organisms for all of the organisms in that population to be happy. Our apartment building can only support 100 people. Any more than 100 is going to cause them to start to be unhappy. So that's what happens when bacteria reach the stationary phase. They can't have any more in the environment. There's not enough room. There's not enough food. The immune system has started to kill them at the same rate that they're dividing. Death equals growth. They are not gaining any more organisms in their population. This number of bacteria remains stationary. So this will remain stationary until one of two things happens. The numbers of bacteria will not increase. One of two things will happen at this point. You will die. You will no longer be able to sustain the fight against the infection, the bacteria, whatever it is. You will die. The organisms will eat you. They will finish their stationary phase out, and then eventually they will run out of food when you are dead. You will die. They will die when they finish eating you, and then eventually they will die as well. They run out of food, and then they hit the death decline phase as they slowly start to starve to death, and then the population will eventually go down to zero. One of three things, I should say. Or two things, I should say. You get better. Either your immune system's going to kick in, or the antibiotic that you started to take is started to kick in. And then they start to die as well. Your immune system started to kill them before they can divide, or the antibiotics started to kill them and control their population. Now, on a petri dish, what's happened here is they run out of food. They ate all of the food on the petri dish. The petri dish only has so much food in it, much like the groceries in your refrigerator. It's only got so much food in it. And unless you replenish the food inside of the refrigerator or give them a new petri dish, they're going to run out of food and starve to death. So every bacteria on the planet adheres to these four phases of growth. The rate, the exponential rate, can be drastically increased, a much sharper curve like this, or a much slower curve, a much more casual, light curve, depending on how fast the bacteria grow. Some species divide quite quickly, which will give you a very sharp curve here, whereas some divide very slowly over time, which will give you a very slow, gradual increase in the number of cells in that population. So the generation time is the number or the amount of time, I should say, it takes for a population of bacteria to double from 1 to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32. You guys get the concept. It's the amount of time that one particular organism takes to double its population. Now that really matters. Some bacteria divide quite quickly. If you have an infection of E. coli or you want to grow them in a lab, you need to know that these guys are going to divide very quickly. You have a limited amount of time to treat your patient. Or they're going to divide quite quickly. You need to have a lot of plates on hand to be able to feed them because they're going to go through a lot of food because they're going to make so many so quickly. Whereas something on the other hand, like Mycobacterium tuberculosis, divides so slowly. Takes almost a couple of months for this thing to divide, a couple of weeks. So you have quite a while before you need to worry about making new plates and things like that. So there are tons and tons and tons of different factors that can affect how quickly bacteria grow, how quickly they divide, and things like that. They're very simple. Quite easily to understand, but they're really important when it comes to setting up a lab or trying to replicate an experiment. And they also help a lot when it comes to understanding how bacteria cause infections in the body. What bacteria require in nature, they're also going to require in the body. And if the body does not have that particular type of things available for it, bacteria at a location, they won't be able to cause an infection there. Say they like a lot of oxygen, they're not going to be able to cause an infection deep inside the muscle tissue. There's not a lot of oxygen there. So we'll talk about this concept a little more in just a second. So one of the main things to talk about when growing bacteria in a lab or causing infections and things like that, or finding bacteria in the environment, are temperature. Temperature is one of the most important things in the world of microbiology. Growing bacteria in a lab usually requires an incubator. And that incubator is designed to replicate the bacteria's optimum growth temperature right here. The temperature that bacteria like, that make them the happiest, where their cells are happy, their cells function. That bacteria is growing, it is dividing as happy as it can, as quickly as it can, as strongly as it can. Whatever temperature that is known as the optimum growth temperature. So if you want to grow bacteria in a lab, you need to figure out what their optimum temperature is and then set your incubator at that particular temperature. So the other two temperatures of concern are the minimum and the maximum temperature. So the minimum temperature is the lowest amount of temperature that a bacteria can grow in. If it's too cold, the bacteria, you guys are the same way. You're going to be really unhappy. You're not going to want to reproduce. You're not going to want to do anything other than just sit there and shiver. And eventually you're going to get so cold that your body's going to shut down. Bacteria are the same way. Eventually they will get to a point where they will not be able to reproduce anymore. They get so cold where they cannot reproduce. Pretty useful if you want to know how to stop them growing in a lab, or help control their spread in a hospital setting or something like that, where you can lower the temperature in a hospital where they cool down and the bacteria might not be as happy. The maximum temperature, on the other hand, is the opposite of that. How hot can it get for bacteria before they're not happy anymore? Once again, could be very useful. If it's too hot for them outside, they won't be able to be found in the desert. That's a nice thing to know if you need to figure out where to research, where to set up a research lab where there are people where there won't be having a risk of becoming infected. Or if the bacteria escapes from the research lab in the middle of the desert, it's not going to be able to cause a problem because the desert will cook it. Or it's also just kind of nice to know, don't set your incubator any lower or any higher than these temperatures. It's also nice to know how fast they grow, because if you need to grow them quickly, you can set it at a very normal temperature, the best temperature, or if you got a little while, you can set it a little cooler and things like that. So I'll show you guys how to read these and what these things mean in just a second. So this is the word form of what I'm about to go over right here. So this is the temperature graph. Bacteria can be broken into 1, 2, 3, 4, 5 different categories when it comes to how they deal with temperature. So I'm going to start right here on this end and work our way towards the other end, the lowest temperature. So over here we have how fast bacteria grow, and down here we have temperature in degrees Celsius. So the very first category of bacteria are something called psychrophiles. Philes meaning lover, something that requires. These guys require a cooler temperature, something that requires it. Philes, requiring. So psychrophiles are bacteria that require a temperature that ranges from about -8 degrees Celsius up to about 17 or 18 degrees Celsius. Anywhere in there they can grow and they will be okay. Their optimum growth temperature can be found right here at the peak of the curve. Minimum growth temperature, maximum growth temperature, and the optimum growth temperature is right there at the middle of the curve. So you can go down there and you can see that's about 12 or 13 degrees Celsius. So if you want to grow a bacteria in your lab that's considered a psychrophile, its classification, you can see here what temperatures you can set your incubator to. And you can also see what temperature that's best to set your incubator to replicate this guy's environment. Now I'm going to skip over Psychrotrophs for a second and move on to mesophiles, but we'll come back to them. So mesophiles, meso meaning middle, phile meaning requires middle temperatures. You guys can see here they range from about 10 degrees Celsius all the way up to about 48 degrees Celsius. Their optimum growth temperature sits at about 37, 38 degrees Celsius. which, if you know anything at all about the medical field, is 98.6 degrees Fahrenheit. So organisms, bacteria that are fall under the mesophyll category classification are organisms that grow best. Their optimum growth temperature is at the same as human body temperature. Not a coincidence. Mesophils tend to be the classification of organisms that cause most of the problems in humans, most of the type of infections. The farther you get away from the optimum growth temperature on this side, the slower they grow. Same over here. The farther you get away from the middle, the slower they grow. The closer you get towards the middle, the faster the organisms grow. Makes fairly easy sense on that one. Thermophils range from about 39 to about 72 degrees Celsius. These guys are going to be found growing a lot in the deserts, growing in hot springs and things like that. Whereas you find your hypothermophiles over here that go from about 65 all the way over the boiling point of 100 to up to about 110 or so degrees in Celsius, which is over 220 degrees Fahrenheit. So these guys are going to be the ones that are found at the very beginning of our slide at that boiling hot springs at the bottom of the ocean and things like that. So if you want to replicate these guys in a lab, you got to have a boiling hot incubator. It's slightly difficult to do. Now, Psychrotrophs, I mentioned I'd come back to these. These guys are technically mesophils, but they don't mind cold temperatures. So kind of just ignore them, be aware they exist. They're a weird classification of mesophils. They can grow in the cold, and they don't mind the warmth either. So these guys, you can put them in the refrigerator, they'll grow fine, or you can leave them out at the room temperature, and they'll also grow fine. Kind of an odd little classification of bacteria. Troph just means liking, not requiring, as Files does. So, nutrients, we've already talked about nutrients. I think that was a messed up slide. So anyway, let's move on to pH. So bacteria in the environment in the world, they are found in environments that have a wide swing of pHs in them. We'll talk about later on in the course that bacteria can be found living in your stomach, which has a pH of around 1 or a little lower, all the way up to your skin, which is basic. They can also be found living at the bottom of the ocean with that negative pH down in there. All the way up to living in highly basic environments, things like soaps and things like that. They can live in that. So they can survive wide ranges of pHs. Now, that's not the same for every species, much like humans can't survive wide ranges of temperatures. We can't survive a -10 forever. We can't survive 200 degrees forever, but some other species can. So it's the same with bacteria. So only certain species can do that. Not all of them can survive every single temperature. Well, anyway, there are some bacteria that really like acid. They require large amounts of acids in their environment. And these guys are known as acidophilic, likers of acid. And you will also find organisms that like basic environments. And these guys are called alkalinophiles. Alkaline meaning basic, philes meaning lover of the base. And you'll see, we'll talk about in the lab section of this course, that certain types of augers and things have been developed that contain different acids and things like that, which will kill off certain species of bacteria that you're not interested in. So if you're looking for a Staphylococcus species that you know can survive in this particular type of acid, you'll find a petri dish that contains that particular type of acid, whereas all the rest of the bacteria that you might find at the same sample of contamination, commensalistic organisms, will be killed, and you'll be left with just what you were looking at. So lots and lots and lots of bacteria can tolerate high presences of salt. The organisms that live on our skin, Staphylococcus, these guys are known as osmotolerant. They can tolerate the high concentrations of salt. Swinging from high concentrations of salt to low concentrations of salt. Osmosis, as you guys know, your body or cells try to dilute the water concentrations to make them equal. So water rushes outside of the staph cells with lots of salt on your skin, and inside the cells with a lot of salt on the inside. So that puts a lot of stress on a cell. So not every cell can survive that. A lot of cells will die. A lot of bacteria species cannot survive this concept of changing salt concentrations very quickly. Organisms that can are known as osmotolerant. Staphylococcus happens to be one of them. We will also find some bacteria. They're slightly rare. They're not so much of concern in the medical field that require high amounts of salt in their environment. They just won't simply be able to live without it. Osmotolerant organisms like Staphylococcus, they can live without high concentrations of salt. You add a lot of salt, they'll also be okay. Halophiles, on the other hand, lover of salt, requirer of salt, they absolutely have to have it, or else they will not be able to grow. So salt, what happens is if you have a healthy salt population on the inside and a healthy salt population on the outside, they're equal. Water will not rush in and out of the cell. The cell will be nice and plumped up, got a lot of water inside of it, stuff can move around inside the cell is happy. Whereas if a cell has a lot of salt on the outside, water is going to rush on the outside of the cell and try to dilute that salt concentration down. And the water on the inside of the cell is left gone. The cell shrivels up, and eventually this will cause the cell to die. This concept is called plasmolysis. Osmotolerant organisms can survive this massive shift in water. Then when the salt concentration drops down, the water will rush back in and fill up the cytoplasm again and cause the cell to become back to normal. Osmotolerant cells can survive this process. Halophiles can survive this process. Most other bacteria species cannot. Once this happens to them, they will die. So one of the main and probably most important, right behind temperature, factors of concern when you're talking about growing bacteria in a lab is the presence or absence of oxygen. So oxygen plays a really, really, really big role when you're talking about growing bacteria. So this also comes into play when, as I mentioned earlier, when you're looking at where bacteria cause infections on the body. Does that site in the body have a lot of oxygen available to it? So if it does, the bacteria that are going to be setting up shop there need or can use oxygen. If the source site does not have a lot of oxygen available to it, the bacteria that are setting up shop there as an infection won't be using oxygen or probably don't need it. One thing to bring up first before I get too far into this is if a bacteria requires oxygen. So not all bacteria require oxygen to live. Some of them cannot even use oxygen. So the bacteria that use oxygen use it the same way that we do. They use it to get rid of their terminal. They use it as their terminal electron acceptor in metabolism. They get rid of toxic byproducts of oxygen. So when we break down oxygen for our metabolism, we build up carbon dioxide. And that carbon dioxide, we breathe it out. It has to be gotten rid of some way, or else it'll build up inside of our bodies and we'll die. Now, bacteria are no different. When they use oxygen, they build up toxic byproducts inside of their cells in the forms of hydrogen peroxide, superoxygen, and things like that. If they don't get rid of those toxic byproducts of their cells, they will die. Now, the problem here is if you are a bacteria that can use oxygen, that requires oxygen to live, you have the enzymes, things like superoxide, demutase, catalase, and things like that, that will break down these toxic byproducts of oxygen into something that's no longer harmful. So you make a toxic byproduct, you have something that breaks that toxic byproduct down, no harm, no foul. Now, unfortunately, if you don't have the ability to break down these toxic byproducts from oxygen, you cannot live in the presence of oxygen. So if you do come in contact with oxygen, you will use oxygen as a terminal electron acceptor. You don't have a choice. Now, unfortunately, what happens is if you use the oxygen as a terminal electron acceptor, you'll build up your toxic byproducts, and then you have no way to get rid of them. So it will build up inside of your cells. You don't have the ability to break down these toxic byproducts, and then these toxic byproducts will eventually kill you. So if you can live in the presence of oxygen, you have the ability to break down those toxic byproducts. If you cannot live in the presence of oxygen, you lack those ability to do that. You don't make enzymes that are required to do such a thing. Now, you can break bacteria up into a bunch of different categories, depending on how they deal with oxygen or how they require oxygen. So the very first classification is something called an aerobe. Aero meaning air, or an obligate or a strict aerobe. So if you're obligated to do something, it means you absolutely have to do it. Strict meaning the same thing, you must do something. So obligate or strict aerobes are organisms that require oxygen in their environment to live. If you want to grow these in a lab, you must give them sufficient supply of oxygen, or else they will not be able to grow. We are obligate aerobes, but we're not bacteria. We don't fall under this classification. But without oxygen, we will die. So these guys, they use oxygen as their metabolism, as their terminal electron acceptor. Without oxygen, they won't be able to get rid of their terminal electrons, which means they won't be able to produce any energy at all. They essentially will starve to death. So the same thing happens to us. We run out of energy, oxygen, excuse me. We cannot get rid of our terminal electrons. We run out of food. We eventually will starve to death and die from lack of oxygen. Your brain can't make the energy that it needs to function, and it just shuts down. So essentially, the same thing happens to these guys. They can't make cellular energy, and they die. They lose the ability to make energy because they cannot get rid of the terminal electrons from metabolism using oxygen. So these guys, they obviously have the ability to get rid of these toxic byproducts from oxygen since they live in a highly oxygen-rich environment. So if you look down here, this is a very common infection, something called a Pseudomonas. This guy it's a very, very, very strict obligate aerobe, requires large amounts of oxygen to be able to grow. And this guy tends to cause problems on places in the body that are very, very, very rich in oxygen. On the skin, underneath the fingernails, respiratory tracts tends to be where it goes. One of the main places that it goes, it causes a lot of infections in the lungs and respiratory tracts, bronchial tubes, and things like that. So places that are available with large amounts of oxygen for this organism to use for its metabolism purposes. You won't see Pseudomonas causing problems deep, deep down in the tissue very often. On the other hand, you'll find an obligate or strict anaerobe. So ana meaning without. So ana are opposite without air. So an obligate or a strict aerobe are organisms that are unable to survive or grow and reproduce in the presence of oxygen. So some of these organisms might be able to survive in the presence of oxygen, but they won't be able to reproduce. They won't be able to grow. They're essentially just going to sit there and do nothing. They're not going to be happy. They can't really function as bacteria. So essentially, they might as well just be dead. Some of these guys will be outright killed by even the smallest amount of oxygen. It just depends on the species. If you fall into this classification, you will not be able to do anything at all other than just sit there or die in the presence of oxygen. So these guys lack the ability to break down these toxic byproducts of oxygen. So what happens is if they come in contact with oxygen at all, they'll bring in that oxygen. They will use it. They don't have a choice. They use it to make energy. They build up these toxic byproducts inside of their cells. It happens very quickly. And then they don't have a way to get rid of those toxic byproducts. The toxic byproducts build up very quickly, and then the organism eventually will die from that. So organisms that are obligates are anaerobes. These guys tend to live at the bottom of lakes and the bottom of rivers, mud puddles, things like that, deep, deep, deep in the ground, in the dirt, where they can shield themselves away from oxygen. So this is a disease down here that you guys probably know as tetanus. Lockjaw brokeback fever is another name for it. It's caused by the bacteria Clostridium tetanii. It's an obligate anaerobe that lives in the soil. A little gram-positive rod organism. So most people think that you get tetanus from stepping on a rusty nail, and that's not technically true. Clostridium tetanii lives in the soil, and where do most rusty nails be when you tend to be when you step on them? They're usually in the dirt. So you step on a rusty nail that's got dirt all over it, and the dirt is the bacteria live inside the little tiny dirt clods to stay away from the oxygen, and you essentially pierce your own skin and shove that bacteria, the little dirt clod on the nail, deep, deep, deep down into your tissues where there's no oxygen at, into your foot, and then they set up shop. And then they can cause problems from there on out. Really horrible disease. Essentially what happens is your body you loses the ability to move its muscles. You essentially just go tense forever, and you starve to death. As you can't eat, your intestines essentially go rigid. You can't pass food through your intestines. Very horrible way to die. You have something called a facultative anaerobe. And these guys are organisms that can grow and survive in the presence of oxygen, and they also can survive without it. So these guys would really like to have oxygen in their environment. They like oxygen. It's a good thing for them to have, but if they don't have it, they have other ways to make energy. So if they got oxygen, it's cool. If they don't have oxygen, it's okay. I mean, I'd much rather have a Ferrari to drive to work, but I'll take a nice Honda or something like that. It's okay. And these guys work the same way. They'll be really functional. They'll reproduce faster. They'll make proteins quicker. They'll be able to do what they need faster in the presence of oxygen. But without it, they'll be okay. They will be able to survive, and without it. A lot of the bacteria that are found in your intestines, a lot of yeast that are found in your intestines, or excuse me, found on the skin and things like that, are facultative anaerobes. You sometimes swallow oxygen, sometimes you don't. So if it's there, they'll use it. If it's not, they prefer to use it. If it's not, they'll be okay without it. So in the middle of all of this, you have the Goldilocks bacteria, microphilic organisms, microaerophiles. So these guys are organisms that require just a little bit of oxygen. Not too much, or else they'll die, and not too little, or else they will die as well. They need just a little bit of oxygen in their environment. These guys tend to like a little more carbon dioxide in their environment, but they have to have just a little bit of oxygen to survive. So these guys tend to find places throughout the body where there's just enough oxygen. These guys tend to be able to move around, so they'll move around inside of the body to find those locations where there's just enough oxygen for them. So, um, overall, you have the the end organism, something called aerotolerant. These guys cannot use oxygen. They don't have the ability to use it. They don't have the ability to break down oxygen at all. They cannot use it for their respiration. They do not care. It does not matter to them. They are considered indifferent to oxygen. They do not use it. They cannot bring it into their cells even if it's there. Most bacteria can. These guys don't, and they won't. So they don't care. If oxygen's there, good. If oxygen's not there, good. Who cares? They don't use it. It does not matter to them. So these guys, they tend to like more carbon dioxide in their environment. So if you want to replicate their growth in a lab, you tend to put them in a CO2 chamber. But it doesn't really matter if oxygen is there or not. So these guys can cause problems throughout the entire body. So strep throat's one of these. You'll have strep throat growing at the back of your throat. It can also grow at different locations throughout the body as well if there's oxygen present or not. So we talked a little bit about organisms that like carbon dioxide. Every single organism on the planet, bacteria included, require some amount of carbon dioxide in their metabolism process, just a little bit, somewhere, some way throughout it. All microbes do. Some of them require more than others. And the organisms that require a ton of it are something called a capthenophile. Now, this is a CO2 chamber down here. Essentially what it is, it's a little box that's got a rubber seal around it to keep the air out. And you put the bacteria on petri dishes inside of it, and you put a little tiny CO2 generator inside of it. It's essentially a little tiny pack that you shake up that's got a carbon dioxide generator inside of it. And it will fill this entire little chamber up with lots and lots and lots of carbon dioxide to make these guys happy. So, how do you figure this stuff out? If somebody handed you a brand new sample of bacteria and said, hey, this is a brand new species, is this guy an aerotolerant, obligate aerobe, obligate anaerobe? What is it? Tell me. How do you figure this out? It's quite simple. The bacteria will tell you themselves. So what you do is you take a new petri dish, or excuse me, a petri dish of bacteria, of pure culture bacteria, and then you take a brand new test tube here with nutrient broth inside of it. Sorry, a nutrient agar inside of it, nutrient gelatin inside of it, solid media. So what's going to happen is the bacteria, once you introduce them into this tube, they are going to move around inside of the tube to find the amount of oxygen that they like. So if you guys know anything about how oxygen dissolves into water or media and things like that, the closer you get towards the oxygen, the more oxygen is going to be dissolved in the surface. The farther away from the surface that you get, the less oxygen that there is. Same kind of concept with the fish tank. Closer to the surface, the more oxygen. The farther down you go, the less oxygen. So, you introduce a brand new sample of bacteria to this sterile test tube. They will orient themselves into the location that's best for them. So over here you see an obligate aerobe. This guy, all of the organisms that are grown are all grouped up at the top of this test tube up here. They're doing their best to get as much oxygen as they possibly can. So you know that this guy cannot, or has to have oxygen. He has to have it because there's nobody down here. If there was growth down here, you'd know he didn't need it. But he has to have it. He's requiring it. He is an obligate anaerobe. He is obligated to use oxygen. Has to have it. Over here, you have a facultative anaerobe. These guys prefer oxygen, but they don't have to have it. So you can see that over here. You see most of them gathered up at the top up here, big blob of them, but they are spread out through the rest of the test tube. Not as many, but they are there. The ones up at the top are getting oxygen. They like it, they prefer it, but they don't have to have it. And that's obvious because there are bacteria growing throughout the rest of the the rest of the tube. Most of them like it. Most of them want it, but they are growing throughout the rest of the tube. That's a facultative anaerobe, bacteria that like oxygen, but they don't have to have it. They prefer it, but they don't have to have it. So an obligated anaerobe, on the other hand, are organisms that don't need oxygen. They don't like it. They can't use it. Stay away from me, please. These guys are going to do their best to get as far away from the oxygen as they can. If they come in contact with it at all, it's probably going to cause them to die. So they're going to be all grouped down at the bottom, sinking, trying to get as far away as they can from that dissolved oxygen. You'll see aerotolerant. These guys can't use oxygen, does not matter to them, who cares? So they're going to be spread out quite evenly throughout this entire tube. You'll find them at the top, at the bottom, at the middle, all the way through at the same concentration. They don't need oxygen, can't use it, does not matter to them if it's there or not. Over here, you'll find a microaerophile. And these guys have oriented themselves in the middle of the tube, just where there's enough oxygen to make them happy. At the top of it, you'll find the maximum amount of oxygen they can survive, and at the bottom, you'll find the minimum amount of oxygen that they can survive. And they'll spend their time moving around in the middle, swimming around, trying to make sure that they stay within those two ranges to make themselves happy. Any more oxygen and they'll die, any less oxygen and they'll die. And that's how you figure out what type of oxygen affiliation that your brand new organism has. You put it in a test tube of nutrient gel, nutrient agar, with a test tube on top and let it incubate. And then come back and look how the bacteria have grown and where they're living at throughout the test tube. So, when you want to grow bacteria in a lab, it's not always super easy. Sometimes bacteria require different things. We've talked about that a lot. You have to give them different environmental conditions, different temperatures, different media, things like that. Some of them require living organisms to grow in or actively dividing cells. Things like viruses will only replicate in actively dividing cells. So depending on what you want to grow in a lab, you have to be aware of that and try to replicate that environment for it. So once you've grown up your bacteria, you've got a pure culture of it. You want to count how many bacteria you have. You want to know what you started with. You want to know what you've got now to see how fast it grows and things like that, to figure out what the generation time is, to figure out how fast this organism is growing in your patient, to say, oh, I've got a while to worry about trying to figure out what this is, or oh, I need to get on this quite quickly. This person's going to die very fast from this. So counting bacteria is pretty easy. There's a couple of different ways to do it, but essentially they're all going to be broken up into two different categories. Something called a direct cell count or something called a viable cell count. So a direct cell count counts every single cell, bacterial cell that's present in a sample, if it is alive or dead. Whereas a viable cell count counts only the cells that are capable of living. Now, this doesn't really seem like much of a difference, but it really does matter. Living cells are capable of dividing. So if it's dividing, it's actively growing. If you have a lot of actively dividing cells, those cells, that infection is still going on, it is getting stronger. Or if it's an environmental sample that you're looking at, the environment's going to become more contaminated and things like that. Um, Direct cell count that counts both alive and dead. Dead cells sometimes matter. Dead cells are when they were alive, they produced toxins. When they die, those toxins are still there. The cells degrade, the toxins will be released after the cell breaks down from being dead. The toxins will be released. So dead cells sometimes do matter. Toxins from those dead cells can cause serious problems. We'll talk about that concept later on. So which type of count you want to do, if it's a direct cell count or a viable cell count, really depends on the information that you're trying to figure out. Or if you're looking at, say, a clinical sample from a patient, or an environmental sample from, say, somebody gave you a sample of water from a creek or a pond or something like that, and your job was to figure out if it's safe for someone to swim in it or not. You need to know how many bacteria are found in it, how much toxins in that water, and things like that. So one of the ways that's the easiest way to count bacteria is something called a direct microbe count. Well, what you're going to do is you're going to take a sample of, say, you've got a gallon of water here, a liter of water from a lake, or a liter, I don't know, say a 5 milliliters of blood or something like that. And you're going to count how much bacteria is inside of that original sample. Now, you can't really count all the bacteria in a liter. That's going to take you forever. Or 5 milliliters of blood might take you a while as well. So what you're going to do is you're going to take a very small measured amount, so maybe a tenth of a milliliter or something out of your liter sample, or a tenth of a milliliter from your blood. And what you're going to do is you're going to see, you guys can see over here, this is a slide that has little tiny grids on it, a microscopic little grid board. And you're going to put that sample over the grid board and put it underneath your microscope. And you're going to physically go in and count 1, 2, 3, 4, 5, 6, 7, 8, every single little bacteria on the lines with your little clicker that you see when the people, that the little buttons that they use their thumb to count the numbers of people that go through lines and stuff. That's what they use to count these things. So the problem is you can't tell a living or a dead organism underneath a microscope. You just can't do it. So is your infection actively growing? Well, you're going to have to wait and find out. So what you do is you count how many bacteria are in a little mill, or tenth of a mill or whatever. And then you do the math to figure out how much bacteria is in a mill, and then how much bacteria is in 5 mils, and then how much bacteria is in a liter. And then you can figure out how much bacteria is in your entire sample, or whatever it was. Just math it back. So the problem here is you need a really large sample to get an accurate estimate of growth. So you do this once, you wait 10 hours, you come back and you sample it again. Are there more bacteria? Are there less bacteria? Then you wait 10 more and you come back and do it again. Are they increasing? Are they decreasing? Things like that. Once again, you can't tell living versus dead underneath a microscope, so your numbers are always going to be flawed. You're going to just be counting the number of overall bacteria. You don't know if they're alive, you don't know if they're dead, and dead things in the counting of bacteria don't really matter so much in the long term of things, unless you're dealing with a bacteria that produces a toxin. So you can count bacteria using something called a flow cytometer. Now this is pretty cool. Essentially what you do is you have a little water bath up at the top, and a little flow of water that's going to flow around it. So you put your sample up at the top in the little well. You guys can see this up here. And essentially what's going to happen is the sample is going to be released very slowly, one single bacteria at a time. One single little bacteria at a time is going to pass down through a little device, a little tube, that has a laser beam that runs through it. That laser beam, every time something breaks the laser beam, will count. 1 bacteria, 2 bacteria, 3 bacteria, 4 bacteria, 5 bacteria. Every single time the laser beam is broken, it will count whatever passes through it. There are tons of different research purposes for this. Blood cells, you can count different types of cells, things like that. Very, very, very useful. Tons of different purposes other than microbiology, but that's one of the ways you can count how many bacteria are in a sample. Once again, you start with a little small sample and do the math to figure out how much was in your original sample. Dead things and living things both break the laser beam. Anything that's physical will break the laser beam. So once again, you don't get just the living cells that are present in your sample. You get both. You get a plate count. Now, plate counts are cool. This is a viable cell count. It only looks at living colonies. Now, this is used a lot for environmental samples when you need to figure out how much water, how much bacteria is, say, in a water sample or things like that. So what you're going to do is you're going to go take a sample from your lake, a big old giant scoop of water from your pond, a big liter, a known amount. And then what you're going to do is you're going to take a small amount, a measured amount, a known amount, and put it on a petri dish. So you start with a liter, and you guys can see here they took 1 to 100. They diluted that liter. They took 1 milliliter, and they diluted it 100 times. And then what they did was they spread that sample out on this petri dish and let it incubate for a couple of hours, and they came back and went to look at it. Well, there's too many bacteria on this petri dish. That's way too many bacteria. I can't count that. That's something called TNTC, too numerous to count. You cannot tell how many bacteria are on that sample. You just can't do it. You cannot get in there and count them all. Now, what they did when they ran into this problem is they had to dilute it again. So 1 to 100 obviously was not enough of a dilution where you could get this. So they diluted it again. 1 milliliter out of our original sample to 1,000 milliliters of water. And that's still too many to count. Still TNTC. You look over here, they did it again. 1 to 10,000. And then 1 to 100,000. And they diluted it even more, all the way down. And what this does is it eventually will allow you to get a very accurate number where you can go in and count 1, 2, 3, 4, 5, 6, 7, 8. There were 30 colonies in my 1 to 10,000 milliliter dilution. Now you have to go back and do the math. 1 to 10,000 was 30. 1 to 1,000 is how many? 1 to 100 is how many? 1 to 10 is how many? 1 to 1 is how many? So eventually you just do the back steps of math to figure out how many were in your original liter sample. This takes forever. It's a very easy way to mess up what you're doing if you're not precise about it. It's very easy to miscount. Lots and lots and lots of problems can be encountered with this. It takes 12 hours per plate for the most part, so I hope your patient has time to wait. But if you're doing environmental samples, they have a time to wait. The environment has a little bit, can wait a couple of days. This is the most sensitive way to do this. Only living cells will be able to make colonies, so you will only see actively dividing, actively growing cells. You will get a very precise number, but it takes a really, really, really long time. It's really easy to mess up with the dilution factors, but the payoff is that you get a really accurate number. So here's the same concept again with the dilution factors. You can look at this one if you want to. What you look at, what you see versus the number of, or the amount of dilution factors and things like that, and how you have to multiply it back up and back down to get the numbers of cells and things like that. You can filter out bacteria. That's pretty easy. You can run them through a large filter, capture them through the filter, and count them that way. Now, unfortunately, filters are also filtering out dead bacteria as well, so if you go to count them underneath a microscope, you're going to get dead bacteria as well. Sometimes it's kind of silly to try to guesstimate or estimate how much bacteria or things like that are in really, really, really large amounts of water and things like that. Really, really, really large samples. And this is kind of the case where it's you kind of just need to know, oh, there's too much here for it to be safe. It doesn't really matter how much is there. You just need to know if it's too much, if it's more than is dangerous. And so anything that's dangerous, it's going to kill you. Anything over that's just overkill. It doesn't really matter how much it is. So it's not really important to figure that out. So what they do in this one is they just do a concept called estimating total biomass. So essentially what they'll do is they'll take a sample of water or something like that. You can see here they've got a big giant algae bloom. So what they'll do is they'll take a sample here, dry it out, take the difference of the weight between the water and the dry weight, and then they can figure out how much weight of that, the remaining weight is all bacteria or algae or whatever that produces the toxin of concern. And if there's too much weight there, they know that there's too much algae, there's too much bacteria, and then it's not safe for people to swim in the water anymore. The too much weight, too much bacteria weight per liter of water, so it's dangerous to humans and things like that. You can count bacteria using a light beam. Some of you guys may have used spectrophotometry in other labs. This is kind of the same concept as a flow cytometer, except it uses a light beam all at once. So there's no flow in this one. It literally just measures the amount of blocked light. An empty tube, you have a light source and a light meter. An empty tube lets 100% of the light from the light bulb go through the empty tube onto our light meter. You add a sample of bacteria into that tube. The light source will be projecting light, and the bacteria that are inside that tube now will deflect some of the light beams and cast shadows. That light beam meter will now register a lower amount of light that is coming through. A little bit of math, some formulas in there, and you can figure out what the ratio of cast light, absorbed light, is to the amount of bacteria that are in that sample. Mass is the easiest way. Just weigh the silly things. You can stick them on and as long as they're in a sample like this, a tube of some sort, you can put them in a centrifuge and spin them around really fast, which causes all the bacteria to be stuck at the bottom of the tube. This is called a bacterial pellet. So you weigh the tube to start with, you spin it around, get the bacteria at the bottom, you pour off the water, and weigh it again. And the remaining difference is the weight of the water, and then you can figure out what minus the weight of the plastic tube itself, and then you can figure out how much bacteria you have per weight. Once again, dead cells have weight, cellular debris has weight, food has weight, waste products have weight. So this is a very, very, very easily manipulatable factor when it comes to relying on this one. Environmental can cause this different heats and things, different temperatures. Maybe your scale's not technically calibrated. So mass is not the easiest, or is the literally easiest way to guesstimate amount of bacteria in a sample, but it's also the most unreliable. So when it comes to bacteria and the relationships that they build with one another in the environment, they're pretty easy. So we talked about growing bacteria in a lab, but sometimes bacteria grow with one another. They do all the time. Bacteria almost always grow with one another, but sometimes bacteria will form relationships with other species of bacteria that benefit one another. Not always. So there's 3 different categories of these. You guys are probably familiar with them. Mutualism, where both organisms depend on one another to exist. Both organisms benefit. This would be the concept of the, I'm trying to think of the clownfish and the sea anemone. Clownfish gets a nice place to live, keeps the sea anemone clean, and the sea anemone gets to be kept clean from the clownfish. Commensalism, where one organism is harmed, where the other organism is not benefited at all. So this would be kind of like a remora fish that just swims around a shark. It doesn't really hurt the shark, doesn't really damage the shark, but the remora fish gets a free ride around the ocean and gets to eat all the shark's food scraps. And then the last one would be parasitism, where one organism is directly benefited, the parasite itself, whereas the host that the parasite's living on is directly harmed. Now, most bacteria are going to be parasites. They don't tend to form friendly relationships with us, but they do sometimes form friendly relationships with one another. So, a couple of the symbioses, the microbial relationships in nature that are found. There's one of these. These are the roots of plants. Bacteria live inside of the roots of plants. This allows them to produce or to fix nitrogen for us, to take nitrogen from the environment and turn it into ammonia. So without the bacteria that live inside of the roots of plants, they would not be able to function. We would also not be able to live. Kleptoplastids, little tiny photosynthetic algae that have been eaten. Tons and tons and tons. The microbial relationship parasite here. This is Plasmodium vivax, malaria that lives inside of a mosquito. It actually will kill the mosquito at some point in time. So lots and lots and lots of different relationships that bacteria and microbial organisms form with different living organisms out there across the world. So Pretty easy on the rest of this. We talked about normal flora already. It's the concept of the bacteria that live on your skin. These guys tend to be your commensalistic organisms. They will form parasitic organisms from time to time, but they mostly just live on you without really causing you any harm. We don't really get a lot of super great benefits from them time to time. They'll help out our immune system and things like that. They don't really do anything to harm us, and we don't really do anything to help them per se, other than kind of give them a place to live. So those are the types of relationships that we form with bacteria. Bacteria that are parasitic, you guys get the idea, the ones that live inside of us that cause disease and things like that when you get sick. You also have bacteria that live in your intestines, that like E. coli, that produce very important enzymes and vitamins things for us, like vitamin K12. So without that type of relationship, they benefits us directly. We would have serious problems. So, bacteria forming relationships with one another in the environment. One of the ways that bacteria talk to one another fairly often in the environment is through the formation of something called a biofilm. Now, biofilms are really easy to understand. Every single surface on the planet is not smooth. There's nothing out there that's really 100% smooth. Everything has little tiny microscopic little surface nicks and abrasions in it. So anything that has liquid or water on it or is moist in any way, shape, or form, like your teeth or the toilet bowl over here or your dog's bowl, anything at all that's moisture, every single wet surface on the planet has a biofilm on it. So what happens? That moisture allows bacteria to move around, to be washed around, to slosh around in the environment. So they'll be washed around in the environment, and eventually one of them is going to stick to that surface. One of those little tiny little abrasions, tiny cuts, tiny scuffs on the surface will allow a bacteria to stick there. So once that bacteria sticks there, it works a lot like the snowball effect. Other bacteria will start to stick to that stuck bacteria. More bacteria start to stick, they build up, they start to grow, they start to divide. They start to film something called a biofilm. Now, biofilms form a kind of a protective layer for the bacteria that live inside of it. They film this kind of a protective, sugary layer on the outside of their cells that keeps them from being washed away, that keeps antibiotics from being able to penetrate down inside of them and things like that. Cells over here on this side of the biofilm can talk to cells over here on this side of the biofilm through a process called quorum sensing. They use hormones and different types of chemicals and things to direct the growth of this biofilm. It's very odd how these things work. They can actually turn off the growth of themselves or say grow slower and things like that. It's very interesting. So eventually what will happen is the biofilm will get so large that it can no longer contain itself. A small piece of it will break off and be released into the environment where it starts to cycle over again. Those little free-swimming bacteria will stick somewhere else and then grow another biofilm. So biofilms are really common on your teeth. It's the formation of plaque. So they grow over your teeth. If you've ever rubbed your tongue across the surface of your teeth and it's been slick, that's the bacteria that grow across the biofilm on your teeth. Down here, plaque is what happens when you don't brush away the biofilm. The biofilm just builds up and builds up and builds up and thickens and thickens and thickens over time. Eventually, it will harden into plaque as that bacteria dies. It just dies and becomes hard into plaque. So that's what a biofilm is. This is also a biofilm. It's not stains on your toilet. It's bacteria that are growing on the little tiny fecal particulates and all the little goodies that are left on the toilet behind when you flush. So this is not fecal material. This is bacteria that are growing on your toilet. So once again, we've reached the point where we will go over our 3 bacterial infections that are you guys are likely to encounter in the world out there in your medical practice. So Lyme disease, very, very, very common bacteria are starting to become a little more common. Not the most common thing on the planet. Very interesting little bacteria. It is a spirochete bacteria. You guys can see that up here. It has a spiral shape. Corkscrew-shaped bacteria, these guys burrow into the bones and the cartilage and things, which makes them very, very, very dangerous. So you get bitten by a tick. This is the stereotypical bullseye rash. The tick bit in the middle there of that little the bullseye in the middle, and then the little circle radiates out. This is not always present. This is given as a diagnostic sign, but it is not a requirement for someone to have Lyme disease. You don't have to have that rash to have Lyme disease. So look for it, but it's not always going to be there. It also might be on the scalp or underneath the hair, hidden somewhere. And it's also not always going to be that big. So the tick bites you. The tick carries the little bacteria around in his intestines, in his stomach, excuse me. The tick regurgitates that bacteria in, or salivary glands, I should say, carries it in its salivary glands. The tick regurgitates the bacteria into the new host. That bacteria sets up shop and starts to divide inside of you. And then causes problems inside of you. It starts to eat cartilage and things like that. So you can get rid of this infection with a doxycycline, amoxicillin treatment, but unfortunately, it can still cause damage. It can leave behind the damage that it's caused. So it causes damage to your body that's not necessarily repaired. Next one is Pseudomonas. We just talked about this one. This is very, very, very common in burn warts. So it's an obligate aerobe. It loves oxygen. Oxygen-rich environments is where it loves to be. It's a gram-negative little rod-shaped bacteria. Very, very, very dangerous infection. Broken skin, lots of oxygen. This is exactly what this thing loves. It tends to grow under bandages on burn warts. So this is what this green is of this person. It also loves to grow in ventilators. Moist, warm, and dark is exactly what this organism loves. Everything inside of this is what this bacteria loves. So it's very easy for ventilators and things to spread around infections in hospitals. So you have to really, really, really thoroughly clean your respirators before you use them on other patients and things like that, your ventilators, because you can easily transmit Pseudomonas infection from place to place. So this guy's super resistant to most antibiotics already, which makes it difficult to treat. So do your best to try to not spread it around. And then our last one, salmonellosis. Not super interesting and is a disease overall. It just causes a lot of intestinal problems. You can survive this one. You have fever, diarrhea, vomiting, and things. Irritable bowel syndrome. About a week or so of the world's worst bathroom issues. It's spread by feces contamination. It's also spread by, you guys can get this, as you know, from eating undercooked, or from eating poultry products, or eggs and things like that, raw eggs, undercooked poultry. It's spread by their feces as well. So undercooked, they get the feces on them at the farm, and they're just not washed or cleaned well. So I think that's pretty much it for this PowerPoint, guys. If you have any questions over this, feel free to email me. If not, you guys take it easy and have a good rest of the day.